Hello, everybody. So I'd also like to thank the organizers for giving me the chance to be here and to you on behalf of the Severe Adverse Effects Consortium and a representative for a large group of people, perhaps mostly go to Chen postdoc actually did all the analysis I'm going to tell you about here. The consortium is who multiple groups, mostly by Mariam who have been created had a it's a remarkable St. Nelson in Saudi. This is as you can really appreciate, bring together a group of cities from to tackle the effects of common drugs and run-of-the-mill headache effects that will send you or me quickly to the hospital. And for it'll take the drug off because we have a shared interest of the adverse way back. Just as we're considering a group of cases, individuals who demonstrate the adverse reaction, comparing them to a group of controls, and we do that by considering their genome differences, specifically focusing so far polymorphic sites have differences in human population, and looking for where we see significant differences in allele frequencies. Now, this strategy has been, and I think the basis for but there are cases when we start looking at severe adverse effects. First off, common diseases are by definition common, whereas severe adverse effects are by definition rare. Then, not rare, the drug wouldn't be bothered anymore. This in turn means what association studies for common traits can amass tens of thousands, sometimes hundreds of thousands of individuals and study, much more limited. The sample sizes that we're be talking are in the dozens or low hundreds. So right off the bat, we're in an underpowered situation here. Worse off, while for understanding common diseases are motivated by high heritability of many, up to 80% some of these traits. For these severe, we often just don't have a family because we don't have enough cases. All that means that while for common diseases, able to, we as the community, to expose very mild effects of genetics on phenotype, our only hope here to expose the very strong signals to severe adverse effects. Now, so the next, what is that the outcome of patient study has limited value in the diagnostic sense to get this increase from 5% to 6% would be with the strongest of variants. Me personally, the main benefit is to understand the disease better research world. If I know that my chances for a severe reaction increased 10-20, that has clinical meaning. So, I'm going to tell you about some of these studies we've been doing. A pilot study of serious skin rash and several studies of drug-induced liver injury. And I'm going to sign off by telling you where we're going now beyond Genomewide association studies. So, serious skin rash, a condition that's really either or both of two separate diagnoses, Stevens-Johnson syndrome or toxic epidermal necrosis. I didn't put up a picture because it's the only ones I was able to find are too disturbing. We're talking about rash from 10 to 100% of skin, so you can get the picture without the visual. There are multiple drugs that have been known to cause this reaction, but this is pretty rare. The only samples we're able to lay our hands on are in the dozens, with the largest sample size for a single drug being 12. And those samples, those are samples that are all with gene ancestry, they come from various centers and collections. If you take the genetic information and look at the principal components of these samples, you appreciate the variability of our samples. To orient you, these samples are from the UK or with ancestry. Someday these samples are from the world champion, and so on. So we tested a million for association to serious skin rash. This is the result. The chromosomes are appear in alternating colors, and the y-axis association signals. So the extent of the probability and what you see here is essentially nothing. And SNPs, this is 1 in a million, and distributed log observed probability tracks the expectation for that measurement. So this is really a null study. We're pretty disappointed, but in fact, the goal of this first study was just to establish the collaboration networks and the sample collection analysis pipeline. We're much more interested in analyzing drug-induced liver injury, which has much more impact on health. This adverse reaction actually accounts for 50% of all acute liver failures. So we're approaching that challenge as well. Again, samples come from all over and we have a somewhat larger size, 400 samples. Parsing that by drug, for at least 2 of the compounds, we have somewhat more samples than we had for all the drugs combined in when studying severe skin rash. So we tackled each of these compounds. The first of which is flucloxacillin, used against gram-negative bacteria. And testing that for association genome-wide, we see what every modern geneticist really wants to see, a very strong peak here of chromosome 6p. The p-value for the associated SNP is less than 10 to the minus 30. And this SNP, RS2395029, is actually known from the literature, has been associated with other traits as well. And it's also known to be a near-perfect correlate, a near-perfect tag for a classical allele of the HLA-B molecule. Specifically tagging the HLB 5701 variant of that molecule. If you look at the distribution of p-values here, this is a lot from the null distribution, but all this deviation is due to this top association and near without that MHC region, and the distribution tracks the null expectation. So we published that a month ago. The top SNP actually increases your risk by a factor of 80. The odds ratio is 80 point something, and it explains more than 80% of cases of drug injury due to flucloxacillin. Went on to consider flucloxacillin is a combination of two compounds, amoxicillin, also penicillin, and clavulanic acid. But you may know this drug under its brand name of Augmentin, a widely prescribed drug. And when conducting the association, we see something that it's to be very similar. A null signal, but very strong association signal, the HLA region. Now, the story here is somewhat more complex. Just zoom in. Just to zoom into this region of between 28 and 34 megabases along the chromosome. The top SNP is genome significant at a p-value of 5 and minus 14. Haven't told you is that the HLA had been as a candidate gene for this to this trait, and a previous report of a classical HLA SNP as a tag, suggesting this is we are the first to show that this is really a association fully explained by the we've uncovered, and not the other way around. When you condition on the when you report the association, our result is still 10 to the minus 4. You can also appreciate that there seem to be multiple signals coming this MHC region for association to cosmoxiclav to formally condition on the top association SNP. A very strong signal, also genome significant at the MHC one region, specifically the HLA gene, which is a new signal. To sum up the picture is that we refer the previous mapped associated signal to a patient just partly but not completely, but means the previously reported association, and we were able to report not at HLA-A. So this is where we are in terms of studies. We're now completing other studies for other drugs that have been known to cause drug-induced liver injury, as well as consider other phenotypes, specifically electrocardiograph-related phenotypes like elongated QT intervals or Torsades syndrome. And to put our work in context, adverse contributors patterns. So we see some of the variants, and we definitely see some of the molecules and pathways. The immune system is association, as well as drug metabolism. So we think a different PGT and mentioned in the previous talk. And these a large fraction of today really mimic the state of affairs also for common diseases. We have a framework in place working and was able to demonstrate several common alleles that show a very strong effects on severe adverse effects, and we're seeing stems coming up again and again. This is what we do today for association to variation. Obviously, the next stage is to try to access variants, and this, the previous speaker had, this needs to be done by sequencing. And the goal is to, when we have different dogs for the specifically looking for genes or actually hit in those severe events, but not elsewhere. So some work to design the optimal set of specifically a constrained budget and controls. How many would you sequence for each scenario? Well, under a wide range of constraints in terms of sample size or effect size you're interested in, you can prove that just 1/E is the correct fraction. You need to sequence more controls because the variant is rarer in controls, so you need more in order to verify that it's not there. Going beyond that, in many of the sequencing technologies available today, one needs to consider a trade-off between sequencing more samples versus investing more in the same set of samples, so increasing the coverage of a few samples. And here the trade-off is somewhat less intuitive. It turns out that there's a wide set of coverages between 8 and 20. But the actual pattern has some kind of there's a good reason for that. This is where we're heading right now. Our efforts, and I think I'll stop here and do questions.
Thank you, Itsik. It's time for questions.
I was wondering if you have looked at any of the, I guess you could call it the metagenomics of the severe cases, such as the gut bacteria, the genomes of the gut bacteria in some of the severe cases. Okay, metagenomics would be definitely interesting to look at, though the samples we have collected are only DNA samples, so we don't have access
to that, unfortunately. Because of the challenges, and so these samples have been collected across continents and decades. So the challenge here is perhaps getting samples in real time in order to do this stuff.
I'm interested in the skin rash case, and there's only a few cases, and it seems is a very common and adverse effect. And do you have any clue that once you have such small sample size, how you might find something interesting?
Okay, so what would we do if we have larger samples for serious skin rash?
Yeah, I mean, in this current situation, you only have a few samples. So if you do not find anything, I mean, important signal. So what? I mean.
So okay, what can we do with very limited sample size?
Yes.
So I think this is part of where we're heading. If the answer is rare mutations that recurrently hit the same gene or the same small set of genes, we have and those mutations are specific to individuals who develop such a reaction, then we'll be able to do that given their entire genome sequences. So I hope this answers that part. But if the answer is not genetic, then we need to address other aspects. Thanks.
So can you explain the sawtooth pattern in power that you showed at the end?
Okay, so why is there a sawtooth pattern for the power of the study when we naively for cases sequence the better we are. So this assuming we're sequencing only cases to population-based controls like a reference set that we're comparing like have been for the colony work. And is that we want to be significant to a particular for a variant seen in some cases and never in 400 European control, and for a particular cutoff, 5 out of to carry a variant if in order to get statistical significance. And the cutoff is required by the number of genes that we're comparing against. We need minus 4. Beyond that, 6, so 6 of 41 is harder to go apart to get for a particular risk model to getting 5 by 40.